Welcome back. Part 3 in this exciting sequence of cellular electrophysiology videos. Here we're going to jump into the world of simulation. You may have done some simulation in the past, various things. It's modeling, computer modeling is another words for it. But the point is we're going to attach equations to the behaviors that came out of the measurements in cellular electrophysiology and then convert that into computer code. Well, we're going to use computer code that a previous grad student of mine created some years ago when he was TAing the class. And we're going to use that code to simulate the behavior of cardiac action potentials using a reasonably sophisticated model, actually one of the early models of cardiac electrophysiology. And we'll get to that shortly. So this again is part 3 of the sequence, but also the third element in this 3-part triangle of theory, simulation, and experiment that we use to explain a lot of things in physiology and certainly in the world of electrophysiology. Now, not surprisingly, given the previous video about the measurements performed by Hodgkin and Huxley, the modeling format, the formalism I mentioned, also shows up that comes from Hodgkin and Huxley. And this diagram really captures the core idea. And if you get this core idea straight, the rest of it hopefully will make sense, because this really is it's arguably the genius that they had and is the core of any model that's created based on their formalism. So what we see here is a very schematic diagram with some intracellular space at the top, extracellular space at the bottom, and in between is the membrane. That's the schematic of the structure we're describing here. Now, inside that membrane, you see an electrical circuit diagram that captures the behavior that we're trying to quantify in this computer model and that Hodgkin and Huxley did in their computer model, because they did carry out calculations, even though the computing facilities were quite primitive, of course, at that time. This is long before modern computers and semiconductors were even available. But nonetheless, we can do that in our laptops within seconds. That's the wonderful world of today. The point is that inside this membrane, you see there is a circuit consisting of 4 parallel channels, so 4 parallel paths through the membrane. Each one of them corresponds to some different element of the structure and function of the membrane itself. On the left, you see the capacitance. Now, capacitance is always present in membranes. It's derived from the physical organization of an intracellular and extracellular space separated by an insulator. The insulator is the membrane, so the membrane is generically an insulator, except for these ion channels. And that also, more than just an insulator, actually contains a dielectric. So the membrane structure, the bilipid layer, creates a dielectric layer. And again, hopefully you've read about dielectrics, know something about them. All they do is increase the capacitance. And we know roughly, it's very consistent across membranes in many living systems, that we have a microfarad per square centimeter as the capacitance for that piece of membrane. And so this model represents a small piece of membrane. Small is variable, it depends on how you set the model up. But this is just a little tiny piece of membrane you can picture here. And that little piece will have a capacitor that's a function of the capacitance multiplied by the surface area of that little piece of membrane. Similarly, the conductances that we see in these other parallel channels are also normalized. So they're also expressed as a function of surface area rather than absolute values of inverse ohms. We have 3 parallel paths, of course, corresponding to the 3 major ion channels that Hodgkin and Huxley first discovered, first exposed in their measurements, and that they'd captured in the model. One of them, of course, corresponds to sodium. One of them, of course, corresponds to potassium. And the last one is a leak. And we'll see this notion of a leak current throughout these models, and very species dependent, but it's sort of an amalgam of a number of other different types of ion channels that are allowing some amount of ions to move, sometimes even different ions sharing the same channel. So the leak is, again, something we dive more deeply in when we want a more complex model. But this basic scenario will already get us a long way into creating the formalism for these models. Now, if we now step back and say, okay, what would the equation be that would describe that circuit? Then we get the expression that you see below. And that is that the total current, so the membrane current in this case, the total membrane current is a sum of all these different paths. We know that parallel paths create a parallel set of currents, and those currents add up together to equal the total current that crosses the membrane. So in this case, we have a capacitive term. That's the term that's dVm dt. So we have a derivative of voltage in this expression. This will require a little bit of work when it comes to the numerics. And then to the right, we have each one of the different equations for that particular ion channel. And these, again, should look familiar because we have Vm, for example, minus Vna. That's the driving force. That's the difference between the membrane voltage and the Nernst voltage for sodium. So again, driving voltage multiplied by conductivity for that particular ion. Replicate that for these three or any number of ion channels that we want to include in our model, and we have an expression. And all that's left to do is solve this whole thing for the value of Vm. Now we'll see this is a differential equation. It has both the derivative with time of membrane voltage and the membrane voltage itself. So we'll need some numerics. Those again we'll describe in the setting of the homework. But the point is that this is a completely sensible, manageable equation that we can actually solve with a computer. And so this is the process, this is the formalism that we use to describe virtually all models that work at this sort of scale, that use this formalism. Now, we've already seen before that cardiac action potentials have different currents, or additional currents, let's say, to those in the nerve cells. And the simplest models came out in the 60s, and there's a whole sequence of those. I won't get into those details. The place we're going to land in history is 1991, and that's the appearance of a model that was constructed by two investigators. One, Luo was the graduate student, Yoram Rudy was the mentor of the grad student. Yoram Rudy is a colleague, actually a friend of mine, who's still alive. He's sort of going into retirement now, but he's had an enormous impact on the field. You'll come across his name again and again. So the Lou-Rudy model, it turns out Lou-Rudy 1, came out in 1991. And here you see the diagram that shows the cell as this yellow blob, the myoplasm. So this is the substance inside the cardiac myocyte. The blue is the extracellular space. And each of those arrows corresponds to a different current. So now we don't just have the sodium and potassium, we have some other currents. In fact, we have several different potassium currents. I mentioned this before, that would happen. We also, of course, have to include a calcium current, and that's what ISI means. That's the slow inward current, which was the jargon name given to calcium, the calcium current in the beginning, because the experimentalists didn't actually know what ion was involved. All they knew was that there was a fast inward current, that was sodium, and then there was this slow inward current, so they called it the slow inward current. And then only later discovered that it was a calcium, largely calcium-mediated current. And then we have IB, which is a background current, similar to the leak current in the Hodgkin-Huxley model. So this formalism has now 6 terms instead of 3, so our equation gets longer with a little more complexity, but we need that complexity to produce anything that looks even vaguely like a realistic action potential from a cardiac cell. And this is the model that you'll implement in the homework, or that you'll use in the homework that's implemented in the MATLAB code. This picture, this diagram here, captures the action potential and the individual currents that drive or feed or produce that action potential. And so below the action potential voltage, you see all the rest are currents. Below that, you see the current, for example, from sodium. You see it's very short, it's very rapid, it's very big, goes up to 300. And you see the units there are normalized by surface area again, so microamps per square centimeter. You also see the other currents that, for example, the slow inward current, I mentioned that was the calcium current that we have. It has a much slower, as the name suggests, time course. It turns on sort of as the sodium is turning on, a little bit delayed, but it turns on less rapidly, so the downstroke is less rapid, and it stays flowing for a great deal longer, virtually the entire duration of the action potential. And of course, that's why one of the reasons the action potential is so long. You also notice it's a much smaller current. So look at the scale here. Here we're getting down to -4 or 4 microamps per square centimeter. And the other currents on the right are the depolarizing currents. The sum of all this produces the action potential, and the code you'll have to play with will reproduce this figure. You'll be able to explore the response in the action potential and the different currents to changes in, for example, ion concentrations and other experiments that I hope you'll actually come up with. So 1991 is a while ago now, and so the models have continued since then. And I'll just mention a couple of, or maybe even one really additional model. That's the Lou-Rudy model from 1994. And you see the same sort of diagram, but a lot more arrows, right? So a lot more complexity. Also inside the myoplasm, you see another region in green here. That's called the sarcoplasmic reticulum. That has an incredibly important function specifically in cardiac cells because it's a storage depot for calcium. And as we'll learn, calcium is incredibly important ion in cardiac cells and in all muscle cells. It regulates or enables contraction to occur. So this addition was important because it allowed for the not the measurement, but the simulation of how calcium moves around, how it's released, how it's pulled up and stored in the sarcoplasmic reticulum, as well as coming across the membrane of the cell itself. And then you also see there are more calcium currents here. There's just a lot more detail. The formalism hasn't changed. That's the thing to keep in mind. All that we have now is additional terms for our equation that adds up all of those currents, which are still flowing in parallel, at least the sarcolemmal ones. It's a little more complicated. We'll have a second set of equations that represent the current flowing in the sarcoplasmic reticular membrane. So that adds another equation that we have to keep track of. But fundamentally, it's the same sort of structure and uses the same numerical approaches. This one also contains those red dots there, some of the pumps involved and the exchangers involved. So you see, we're just adding more and more realism to the model to hopefully not only describe action potentials more accurately, but give us a more powerful tool to explore what the response of the cell would be to changes in mutations. For example, there's a whole world of what are called channelopathies. These are mutated genes in certain unfortunate people that can create abnormal behaviors of some of these more subtle currents, the potassium currents, for example. And we think that's the underlying cause of a number of arrhythmias, for example. Another major thrust here would be to introduce drugs into the story, drugs to hopefully control in a positive way or help patients who have abnormal heart function. And we need to know how those drugs interact with the channels and describe their behaviors. So the models have lots and lots of utility. They also form a core element of the models we'll see later that hopefully describe the tissue or the whole heart itself. So these kind of models will not disappear as we move into larger scales. They'll just be now part of an even more complex story. So there's a number of these models. There's a whole history of these. Again, we have other courses that go into the details of these. But suffice it to say that these models really are very worthwhile, very useful in the world of electrophysiology and in cardiology more generally. So you will do a homework assignment that enables you to play with the 1991 Lou-Rudy model and explore and experiment with it to see how it behaves. The last thing I want to talk about briefly here is the control of the heart rate. So I mentioned control is always important in physiology, and we need to know the mechanisms that allow control to occur. And we'll see that across scales there are different mechanisms. There obviously have to be feedback mechanisms, negative feedback, maintain homeostasis, all those sorts of things. But it turns out heart rate is controlled at the cellular level, and so I thought this was a good place to bring it in. Now it's in the cells, a specific subtype of cells we'll learn more about, that actually are what are called the pacemaker cells of the heart. Now, most cells in the heart, if left to their own devices, left unstimulated, will simply sit at rest forever, as long as there's enough material to keep them metabolically alive. But there are some specialized cells in the heart that spontaneously generate their own action potentials. They don't need to be stimulated. These are pacemaker cells, the most important of which are in a node, a region of tissue called the sinoatrial node that we'll also hear more about. So in that sinoatrial node, we have action potentials that look different from what you've seen before. You can see that this SA nodal action potential rises much more slowly. It doesn't have that steep action potential, but more importantly, you see that it's never truly at rest. Even when the potential goes down to its most negative value, it's then drifting upwards. And that's what makes it a pacemaker cell. So that potential, that depolarizing potential, will happen in the background by itself. You can take a single cell, extract it from a heart, and put it in a bath, and it will show this kind of behavior. So this cell will beat spontaneously like a clock over time for the life of the animal or the human. And there are different currents involved. as we say, whenever you want to understand action potential shape, the first question to ask is which currents are involved and what shape do they have? And here we see that the current that carries the upstroke is no longer sodium. That's the biggest difference here. Sodium channels don't really exist in the sinoatrial pacemaker cells. Instead, we have calcium. A couple of different types of calcium, subtypes, but they elevate the potential first slowly, that slowly rising potential, and then once a threshold is reached, the more rapid calcium potential, and that causes the action potential to fire. And then we have potassium that restores the potential back to normal, and then we start again. So that IF is what's called the funny current. Who said electrophysiologists don't have a sense of humor? This is along the same time as the slower inward current was first identified. So this is the funny current, and the funny current is there to constantly depolarize the membrane potential of these specialized cells. So this is how we have a heartbeat. This is why our heart beat in a regular way. And this is why transplanted hearts will beat in a regular way, even when they're not connected to the nervous system of our bodies. Now, how does that get regulated? Here's the tracing showing the sinoatrial node, or schematically sinoatrial node action potentials, a couple of these action potentials. It turns out that there are two neurotransmitters you've certainly heard of before, epinephrine, norepinephrine, and acetylcholine. And it turns out these two neurotransmitters directly affect the membrane. They affect the conductivity of the currents, and they affect the potentials. So let's look first at the role of epinephrine. So epinephrine increases that funny current, that IF. It regulates it up. And then we know something about the system. There's a beta-1 receptor system that is involved in this. But the important part for us is it changes the rate of rise of that funny current. And of course, when the rate of rise goes up, as you see in this red dashed line, then the action potential reaches its threshold earlier and then fires its action potential earlier. And this way we increase the heart rate. The opposite happens with acetylcholine for a slightly different reason. So in this case, acetylcholine changes the potassium conductivity, the background potassium conductivity or permeability, and that drives the membrane, the lower minimal membrane, we don't call it resting potential anymore, but this most negative, most polarized potential into a more negative range. So it drives, you see in this purple tracing here, that lowest potential to a more negative value, so that even if the funny current is flowing, and let's say it's flowing at the same rate as the unmodified action potential, it will just take more time to reach threshold because it's starting from a lower value. And then this is how acetylcholine can regulate the rate of the heart to a slower value. It reduce the rate, increase the period, slow down the heart rate. So here we can see how a set of neurotransmitters that, of course, you know are associated, or maybe will know soon, are associated with the sympathetic and parasympathetic arms of the autonomic nervous system, how these two neurotransmitters can directly affect the cell, and that cell, because it is a pacemaker cell, ultimately determines the heart rate of the entire heart. And so again, we see physiology is tricky, and that these effects can play out at different scales and yet can cross scales in terms of their influence. So here's an example again, cellular mechanism that's controlling the entire heart. There are other mechanisms like it. There are other mechanisms that we'll see will play out at different scales, and we'll get back to some of those in the next few weeks, actually. So hope that was informative. Hope you're totally psyched now to go play with the computer models. So check out the homework. The instructions are all there, as well as some more background information on the Hodgkin-Huxley formalism, how it's created, even some of the numerics of how the code is implemented, if you're curious about that. And hopefully you'll be able to, like I say, carry out some in silico experiments and learn something about how cardiac membranes behave. So with that, I'll wrap it up. Look forward to hearing your questions, and thanks so much for your attention. Bye for now.